Greetings and salutations, magical chemists. Let's talk about the glories of spectroscopy. Spectroscopy officially is a broad term, and it's a study of the interaction between matter and electromagnetic radiation. Almost all the information that you've been learning about the structure of atoms, where their electrons are located, it's all determined by spectroscopy. So there are all sorts of things you can use spectroscopy for, and there are so many different kinds. You can determine what atoms or elements are in a molecule, the structure of molecules, or just the structure of any given substance. So here is a brief or moderately brief summary of all the different types of spectroscopy. The first one is one you probably have never heard of before. It is not that common in chemistry, but it is the AP test really focuses on this one thing called Photoelectron Spectroscopy, or PES. So we are going to talk about this in more detail later, but it's using X-ray radiation, so high energy, and it's kicking out electrons. And it can therefore determine not only what element you have, but the location and number of each electron in the atom. So PES, super awesome. There's also something called UV-Vis, which is UV visible light spectroscopy. And so this is also high energy. So UV-Vis spectroscopy can use either ultraviolet light, which of course we can't see, or it can use visible light. When it uses visible light, then you might have heard of it before. It's called colorimetry. So what you can do is in either using ultraviolet or visible light, you're basically exciting electrons. If you provide enough energy, they jump out of their ground state to a higher energy level. When they fall back, or just from the energy absorbed, you can identify what you have, or even you can identify the concentration of solution. There is something called infrared or IR spectroscopy. This is vibrational spectroscopy. You'd be amazed by how much information you can discover by causing molecules to vibrate in different ways. It has to do with what bonds they have, who they're bonded to, and so there's actually tons of information that you can find out. There's even microwave spectroscopy. Now, microwaves, of course, are pretty low energy, so this causes atoms to rotate. It's very low energy, but you can actually do something pretty dang helpful. You can determine where the hydrogens are located. Sounds like a small detail, but there's a lot of hydrogens in a lot of different things, and if you know where the hydrogens are, you can figure out where other stuff is. So, in short, tons of different types of spectroscopy. So, let's go ahead and take a look at an absorption spectrum for a given sample. So in this case, notice there's part that's occurring in the UV visible spectrum, and this is what's happening with your electronic transitions. There's part occurring in the infrared part of the spectrum. This is where the molecular vibrations are occurring. And there's parts in the microwave part of the spectrum, which is where the rotations are occurring. So I have two questions for you. The first one is based on that picture, and the second one is just based on what we've done in class together. So I would like you to take a moment and answer these delicious questions, and when you're ready to check the answers, unpause the video. Okay, well, if you're going to rank them in order from least to most energy required, clearly rotations must be at the lowest energy, because they can do that with microwaves, and microwaves are very low energy. Which means the one that's kind of the energy in the middle must be those vibrations, because that's what we're doing with infrared radiation, which is higher energy than microwaves, but not as high as the rest. And that means, of course, it must be those electronic transitions, forcing those electrons to jump up to a higher energy state. That requires the most energy, because that occurs with visible and UV radiation. And for the second one, I was hoping you'd remember that we have indeed done colorimetry before, which is officially UV-Vis spectroscopy. This was back in kinetics. We did the crystal violet lab. If you remember, we were able to use the colorimeter to study the changing concentration of the crystal violet, because the crystal violet was a really pretty, violet color. And as it decreased in concentration, then more and more of the laser would pass through it. So we used this to determine the concentration of a colorful solution. All right, let's turn to the next page and talk about atomic absorption and emission spectra using that UV visible radiation. You might remember atomic absorption emission spectra from pre-AP. It's looking at the relationship of atoms, their two states relative to the amount of energy absorbed by their electrons. In the ground state, of course, this is when electrons are in their lowest energy state. So lithium, there's a nice, small, simple atom, only 3 electrons. In the ground state, lowest energy, the first 2 electrons, Aufbau principle, are going to go into the lowest energy orbital available. And then the next one, of course, the last one, the third, will go into the 2s energy level and sublevel, because that's the next place it has room to accept it. However, we also, if you add energy, you can make an electron jump up out of order into a higher state. Now, technically, any of these 3 electrons can jump up, but I left the 1s electrons alone, and I said that 2s electron gained enough energy to jump up to 2p. So, it could have jumped all the way up to 4p or 7s. That's the thing. It depends on the amount of energy they absorb. But the idea is you will recognize an excited state when you see it because the electrons are out of order. So atomic absorption spectra can be used to determine the concentration of elements in a sample by forcing electrons to jump up when they absorb a specific amount of light. As the electron moves from its ground state to an excited state, of course it has gained energy, so the atom's overall potential energy increases. And so you can look at the absorption spectrum to see the relative intensity of each frequency that has basically been absorbed by the electrons jumping up. This is sometimes called a dark line spectrum because you've got the whole rainbow of colors, and you'll notice there's a dark line everywhere that energy was absorbed because there was a specific electronic transition. And so the dark line spectrum is going to look different depending on each atom because they all have a different number of protons, so their electron energy levels have slightly different energies and thus require slightly different amount of energies to jump upward to a higher energy level. So you can identify what elements are present by looking at the presence and location of the dark lines. Now, the opposite of a dark line spectrum, the absorption spectrum, is an emission spectrum, the bright line spectrum. This is also used to determine the identities of unknowns present in a given sample. But this time, it's when you have excited electrons falling back down to ground state. Well, when they do so, they release the energy they had gained or emit it. And so you look at the emission spectrum. And it's funny, the emission spectrum is just going to have bright lines everywhere there were dark lines on the dark line spectrum, because the exact same frequency of energy that was absorbed is now being released when those electrons drop back down. So let's go ahead and try a practice problem using an emission spectrum. I guess actually this is an emission spectra because it's a lot of them together. Notice it's giving you a whole bunch of different elements and the locations of their particular electronic emissions. And then there's an unknown. Your job is to figure out what the heck is your unknown. Well, try it. And when you are ready to check your answer, unpause the video. Did you realize it was lithium? In this case, you'll notice the pattern of lines of your unknown exactly matches the pattern of lines for lithium. So their emission spectrum match. That means they must be the same element. All right, if you have not yet, go ahead and try questions 2 and question 3. When you're ready to check, unpause the video. For question number 2, notice it says absorption. So we want something jumping up, which means it sure as heck, if you're absorbing energy, you're jumping to a higher energy level. But we want something that would have the lowest amount of energy being absorbed, which means this is where you have to remember that as you get further from the nucleus, your main energy levels get closer and closer together. Thus, they require less and less energy for something to basically transfer between them. So in this case, the lowest energy absorption is going to be the transition furthest from the nucleus, but still jumping up because you absorbed energy. Now, for number 3, we want fluorine, but we want to have fluorine in an excited state. Fluorine, if you look at your periodic table, has 9 total electrons. Well, this one is ground state fluorine. It has 9 total electrons and it's in its lowest energy state. But if it's in the ground state, it can't be that one. So It also can't be this one because it only has 8 electrons. But it can't be D either because that one has 10 electrons. Which means B is totally flowing in its excited state. Notice, one of the 2p electrons jumped up to a higher energy level. All right, we are ready to talk about a new and exciting type of spectroscopy, Photoelectron Spectroscopy, abbreviated PES for short. So, Photoelectron spectroscopy is a technique we use to determine the ionization energy, sometimes called the binding energy, of every single electron in an atom. You may recall back in the day from periodic trends, ionization energy is just the energy required to remove or ionize an electron from an atom. Remember, sometimes in photoelectron spectroscopy they will call it the binding energy, but it means the same thing. So the units of binding energy are often kilojoules per mole or even megajoules per mole because some of those electrons takes a lot of energy to remove them. And, okay, this is super weird. Binding energy is going to be, so it's going to look like a graph, and it's on the horizontal axis, but energy is usually decreasing from left to right, not increasing like normal people do. However, that's not always true. Sometimes it's the normal way, so you just got to make sure you're watching your axes. Otherwise, you will get everything backwards, and we shall cry together. All right. So what's happening in photoelectron spectroscopy is you are basically smacking an atom with light. That's pretty much all of spectroscopy. So you've got some kind of incoming electromagnetic radiation, doesn't necessarily have to be visible, and it's going to come in, and it is basically going to kick an electron out. Boom, escape. Now that electron will have some leftover kinetic energy. Basically, the energy of the light coming in has to be the energy it took to kick off the electron plus the kinetic energy. So what we do is we basically are measuring the kinetic energy and using that to determine the binding energy. That's, yay, simple math. It's not too bad. So let's talk about how you would interpret a photoelectron spectrum. There are 2 things you want to look at, the peak height and the peak location. Peak height corresponds to the relative number of electrons in each sublevel, whereas the location, this is the part what's being plotted on the x-axis, refers to the relative amount of energy it took to kick out those electrons. So the peaks at higher energies are the sublevels that are closer to the nucleus, so they're most attracted and take the most energy to remove. Which means things at lower energies, yeah, that's a typo, must be the sublevels farther from the nucleus. Now, you will often be expected to compare the photoelectron spectrums of different atoms. The idea is, as the number of protons in the nucleus goes up, the binding energy will also go up. This is straight up Coulomb's law. Greater magnitude of charges, greater attraction, greater energy to separate them. All right, now, as the number of electrons in a specific sublevel increases, the height of the peak is also going to go up. Because remember, the height of the peak tells you how many electrons there are. More electrons, taller peak. Okay, I know this is super abstract, so let's look at an example, because I think when you see the example, things will start to make more sense. Okay, so here's our example spectrum, and what we want to be able to do. And honestly, the first thing you should do, regardless of what you're doing with your PES spectrum, is you're going to label each peak. Which energy level and sublevel? Like, is it 1s, 2s, 2p? And it's always going to start with 1s. But the thing you have to be careful about is you have to look at the energies. Notice, this is that weird thing I was talking about. The highest energy is closest to the origin. It's so weird. Which of course means the energy is getting smaller and smaller as you go further to the right. The important thing to remember is that the higher energy means these are the ones that are hardest to remove, so they must be closest to the nucleus. Well, what could be closer to the nucleus than the 1s sublevel? And that's why the highest energy peak on your PES spectrum will always be the 1s sublevel. And if you have any electrons afterwards, which we do, clearly, we've got lots more, this means there's always going to be 2 electrons in that sublevel. So this relative height must be equal to 2 electrons. Well, if that's the height that means 2 electrons, well, notice, 1s closest to the nucleus takes the most energy to remove. The next one must also have 2 electrons in it, and what comes next after the 1s? Hey, look, it's 2s, and it's got 2 electrons as well based on the height. Hopefully you realize that that means the next peak must be 2p. And of course, the maximum that 2p can have in it is 6 electrons. You know that it has all 6 because there's more electrons afterwards. We're assuming for Vis spectrum it's always going to be in the ground state. They should never be jumped up out of order, which makes this even more straightforward. Because after 2p, well, it must be 3s. And that must be full as well because there are more electrons after. Now, after 3s, of course, is 3p. But here, hopefully you can tell, it is definitely not full. It did not make it up to the height of 6 electrons. So this is where we're going to do some very kind of rule of thumb measurement. You basically look at the height of this, and then you'll be like, hey, okay, if this is 2, I have 1, 2. It's twice as high. Ta-da! So if that one was 2 electrons, this one must be 4 electrons. Because if it was 3 electrons, then it only should have been somewhere around here. So it's funny. You get a quantitative answer, but at the same time, you're kind of like, is it twice as much? Is it, 1.5 times as much? And that's what you're figuring out. But now, that makes this super easy to identify this, because it has filled in all of its orbitals except for the 3p energy level and sublevel, and that one only has 4 electrons in it. So if you check your periodic table, well, 3p4 must be sulfur. And so part C just says, well, which electrons would be the first to be removed? So this would be like classic ionization energy. Well, remember, the first electrons to be removed are going to be the easiest ones, the ones that take less energy. So the very last ones to be filled are the first ones to be removed. So 3p electrons must be the ones that are removed first, which means the last electrons would be the hardest, the ones that are most attracted and closest to the nucleus. So those, of course, would be the 1s electrons. And that's it. That's how you read a photoelectron spectrum. Let's look at one where that gets a little weirder. The concept is still the same, but I don't like the way this is graphed. But they graph them like this, so it is what it is. Okay, notice a couple things here. First, you've got 3 different rows. They're basically saying it was too big. We couldn't fit it on one paper, so we put 3 different rows, one on top of each other. Then we have to look at the energies. Okay, this one actually does have the lowest energy on the left. But as we go up and are going across, each one is higher and higher. So technically, the lower energy is in the very bottom on the left, and then the highest energy is all the way on this top. So now, once you know where the highest energy is, you recognize, well, highest energy, and look, it even tells you, how nice of them, there are 2 electrons here. So this must be the 1s sublevel with 2 electrons inside. Then, there aren't even any peaks on the second one, so I don't even know. Why did they put that there? I have no idea. But it's okay. We're just going to ignore it and move on to the next one. But be careful. After 1s, we need the next highest energy. So notice, the higher energy is over on the right this time. So that means this lovely little peak over here must be your 2s electron, your 2s sublevel with 2 electrons in it. So next, after 2s, of course, would come 2p. And hey, look, it's got all 6 electrons. And then last but not least, the lowest energy peak that we have, this must be the highest energy sublevel. And the next one is 3s, and they told us it has 2 electrons inside. So this one is a little harder to read, but as long as you follow the energy high to low, you'll always be able to fill in the electron configuration. Which means, since this ends at 3s2, this must be magnesium, which has 2 valence electrons. Now, take a moment, if you have not, and try its noble gas configuration, just for fun. When you're ready to check your answer, unpause the video. And there you have it. It was basically magnesium is full all the way to the second energy level, so that's neon, and then we only had the valence electrons afterwards. All right, let's look at one more example, and then we will be done. Well, this one is a fun one. I would like you to try labeling each one, just like we've been doing. Where's the 1s, where's the 2s, so on and so forth. So take a moment to label each peak, and when you are ready to check the answer, unpause the video. All right, how did you do? Hopefully this is starting to make sense. So, did you recognize this one? This was sulfur. So we actually looked at the spectrum before, but it was convenient to try it again. So now what we're going to do, a little bit sneakier, let's add in, I'm using a different color. Let's see, for oxygen, oxygen's totally red. Why not? We're going to sketch in the relative locations and peak heights for the spectrum of oxygen. Now, you don't have to know exactly where they are but you do have to know for each peak, is it going to be at a higher energy or lower energy? So remember, it has to do with number of protons. So first, just go ahead and try to figure out where should the 1s peak be for oxygen. When you're ready to check your answer, unpause the video. Okay, well, to figure this out, you had to think about number of protons. Remember, sulfur's got 16 protons, but oxygen only has 8, which means sulfur's nucleus is just objectively more attractive. Coulomb's law, right? Greater magnitude of charge, greater attraction. So oxygen's 1s peak, it'll be the same height, it has the same number of electrons, but it's going to be less energy, so it's got to be further that way. Where? I don't know, a little bit somewhere over there. All right, now that you have that, take a moment if you have not already, draw in the rest of the spectrum for oxygen. And when you're ready to check your answer, unpause the video. All right, how did you do? Did you recognize that the spectrum for oxygen will not have as many peaks? Because oxygen, of course, only goes up to 2p4. So it will have its 2s peak, same height as the 2s peak for sulfur. But its 2p peak will be shorter. There are only 4 electrons inside. All right, now that we have that information, we can do our very last problem. Because it says, does it require more or less energy to remove a 2p electron from oxygen compared to a 2p electron for sulfur? And hey, justify in terms of Coulomb's law. So take a moment. I would like you to totally try this question. And then, when you're ready to check your answer, unpause the video. Did you realize it must take less energy? I mean, if you figured out where the peak was, of course, look at that. The peak is lower in energy, so it's going to take less energy than it would to remove the comparable peak from sulfur. Of course, the key is explaining that. Well, with Coulomb's law, we're coming back to the magnitude of the charges. Remember, energy of attraction is directly proportional to the magnitude of the charges. If you have less protons, less charge, less attraction. Thus, less energy required to move the electron. And there you have it. Notice, if it says justify in terms of Coulomb's law, you sure as heck better mention Coulomb's law. All right, I will see you in class for fun and excitement with spectroscopy.